discussing how H. pylori infections come to be and why they're so persistent. And we'll be looking at a bit about the treatment options available, diagnosis options available as well. So H. pylori is an infection that damages mostly the stomach lining, and the way it does that is it reduces the ability of the stomach to digest, and that is this bacteria here has the ability to make the stomach acid less effective. That is by raising its pH. Usually our pH is very low. Humans have traditionally uh, about 1.5 to 3 pH, which is lower than even like a lion, let's say, more like a vulture. But when H. pylori attacks, this could be raised up to 5, which is no good, right? Because you need to digest meats, you need to digest stuff with proteins, and although the actual absorption occurs in the small intestine, the stomach is very important in terms of breaking down the macromolecules in smaller chunks, so it's easier for the small intestine to get into and for the enzymes to attack. Uh, so this is the general scheme of things. The pancreas and liver, those are accessory kind of organs where they give bile or other enzymes for the pancreas, whose names are escaping me right now, but like that are important for small intestine digestion, but also stomach digestion. The appendix, as we all know, is a useless organ, or so they say. The rectum, the anus, but uh, today we'll be looking mainly at the stomach. So the stomach, it's a muscle, and it's also a digestive kind of organ. So for that reason, it has mechanical digestion and chemical digestion. And this time, with the H. pylori infection, the chemical aspect of it is significantly interfered with. And so they did a study with patients who have gastric ulcers, and they found their microbiota is up to 94% H. pylori, whereas in healthy patients, that number is way lower. And there's a lot more diversity in terms of the microbiota of the gut. So this is called dysbiosis, which results as a result of H. pylori infection. This definitely does disrupt uh, the energy levels of the body. It also results in inflammation. It interferes with your ability to absorb a lot of important macromolecules as well as vitamins. Now, interesting enough, Africa does have one of the highest H. pylori incidences, but they also have a very low gastric cancer rate. So they call this the African enigma. But in general, a higher uh, incidence of H. pylori infection is also related to higher gastric cancer rates, except weirdly enough in Africa. Now, in terms of treatment, antibiotics are the main go-to, and that will be a PPI or a proton pump inhibitor, and also three different antibiotics, usually clatromycin, metronidazole, and amoxicillin, maybe. Now you could add on other stuff to it. For example, traditional Chinese medicine is antimicrobial, so it could be working against the H. pylori infection itself, and it also does help the anti-inflammatory properties. So that would be like compounds like berberine that are important in traditional Chinese medicine, and there's other compounds too. But I'm not really a traditional Chinese medicine expert, so you're gonna have to do your own research there. Probiotics, as we saw, like the lack of diversity may be related to why H. pylori is so devastating in terms of gastritis and gastric cancer. Um, Now, for the triple therapy, you have to be careful because some areas do have high antibiotic resistance. For instance, Japan has high resistance to clatromycin in general. Maybe this is because of their overprescription or just the different strains of H. pylori over there. So, in that case, then you would use a different method. I think I have it somewhere down there. This is just a description of what the typical stomach pH is. It's 2. The intestine is 7. the body does a lot to neutralize a lot of the acid as it enters the duodenum. Now, if you lack the ability to neutralize, then you might get ulcers down there too. Duodenal ulcers, they're called. Now, it's very hard to deliver drugs in general in the low stomach pH because the low stomach pH degrades uh, due to the acidity a lot of medicines that might be trying to get in there. And amoxicillin is no different. It does break down in the acidic pH. So why is our stomach so acidic anyway? Well, we humans is hypothesized evolved eating carrion, like scavengers, such as hyenas or vultures. So after a lion or a wolf kills the deer or the moose or the elk, we chase it off and we take its food. Now, this is called carrion, and carrion has a higher level of parasites and bacteria. For that reason, to eat carrion, you need to have very high stomach acid. But bacteria can't live in this environment. So, how does H. pylori do it? We have very acidic stomach pH, but H. pylori just gets in there and makes it its home. How does it do that? It does it by pissing all over the place. I just found this funny. I mean, it's kind of accurate too. So basically, it uses urea. It has an enzyme called urease, which turns urea into carbon dioxide and ammonia. Before I get ahead of myself, let me just show you here. So it turns urea into ammonia and carbon dioxide. So urea is already pre-existing metabolite in our body. We have lots of urea anyways. It's a waste product of a lot of amino acids. Now this urea could be turned to ammonia. And the way it does that is that's a more acidic version of it. Basically, is by the enzyme urease, which the Helicobacter pylori bacteria has in abundance. This enzyme then converts the urea to ammonia. And if you notice, urea has NH2, ammonia has NH3. That means there's three hydrogens in ammonia, but only two in urea. So what does that mean? Where did the extra hydrogen come from? That comes from the existing stomach acid all around it. That means we stole a proton from the stomach acid and put it in the non accessible ammonia. So, what is a proton? A proton is acidic, an H plus, right? We took some acid and put it away in ammonia. We just basically shelved it. So that means now our pH has increased. It used to be a pH of 1 or 3, very acidic. No bacteria can survive there. But now it's going to 5 to 7, maybe 6, 7. And for that reason, then you say, well, H. pylori has made your stomach less acidic, has made it a higher, more alkaline pH. Now, this is helpful in terms of diagnosis. So we have a more acidic stomach acid than a lion. Even though a lion eats lots of meat, so their stomach is very acidic too. We have a stomach acid that's similar to a vulture, although theirs are a little more acidic, a lot more protective against bacterial infections because they do eat very rotten meat, you know, that we can't handle, I think, hopefully. So this is the triple therapy that's often recommended: a metronidazole, clatromycin, and amoxicillin. And I know I'm butchering these names, so please uh, be nice. Problems: antibiotic resistance, big problem nowadays, especially in areas with overprescription where uh, agricultural runoffs contain antibiotics. So the environmental strains that exist already have the antibiotic resistance built in or selected for. So in those areas with high clatromycin resistance, physicians have some success was using vonoprazan at 20 milligrams and amoxicillin at 750 milligrams. So those are in areas with high clatromycin. But anyways, the doctor will figure out a way to get around those resistances, but they should be very watchful for those resistances in the first place, because if you ignore them, then the therapy is not going to work for the patient. And this therapy is very necessary because if you let it occur over and over again, there's a high chance of gastric cancer, as we saw. So what's antibiotic resistance anyway? Maybe I should mention this. So let's say those orange ones are bacteria that don't have resistance, meaning they can't survive against a medicine you give them. Let's say the medicine is, although it doesn't really, I don't know if it's antibiotic, but Tylenol, because everybody knows what Tylenol is. These orange ones, uh, they're giving you a headache, and they can't survive against Tylenol. So when you take a Tylenol, they die. But this blue one, it can survive against Tylenol. So it survives and it stays. And now it has all this empty space to kind of grow more of itself. So what you've done is you've effectively selected for a resistant strain to survive and grow in numbers. Now, some bacteria have the ability because they have the, what is it? Some kind of conjugation, I think it's called, where they could give the gene for resistance to another bacteria, maybe completely unrelated in terms of strain. Then the resistance can be horizontally transferred that way. So then you get into a really big problem when not only the bacteria and its descendants, but other neighbors could also develop the resistance. Then that's going to be a huge problem in terms of a population level, uh, epidemic response you may try. So how do you diagnose H. pylori infection anyway? Well, you could just use antibodies to detect it in the serum, meaning blood, saliva, or urine, pee. Now, what you pee out is what has already been in your blood, so you should be having a fairly accurate picture. One problem is, let's say the infection was cured and the H. pylori is no longer there, the antibodies might still be present in the blood. So it might not be an actual indication of an ongoing infection. What we call a false positive may result with this serological test. A sero just means blood, I believe, and a test, you know what a test is. Okay, so then there's the urea breath test too that you could use where, because as we said, urea smells like pee, I think ammonia, the smell of ammonia. So you could kind of try to sense that and you could just smell the breath and try to identify it. Or I think they use a more sophisticated version. They use isotopes, so they give them an isotope filled. Isotope means just a unique type. Let's say the carbon instead of a carbon 12, you give them a carbon that weighs 13 atomic units. That's what an isotope is. So an isotope, unique urea, which is separate than what is produced in the body, and then you try to capture the carbon dioxide and see if it is being broken down inside the body into ammonia and carbon dioxide. If it is being broken down into ammonia and carbon dioxide, then you have your evidence that the urease enzyme is present. Now you could also use a PCR test to detect the H. pylori DNA. Another thing you could do is you could take a biopsy. Uh, Biopsy will let you take a sample tissue, put it into the pH test, and if the pH test shows an alkaline, then you have H. pylori because your pH is way too high, meaning your stomach acid is not acidic anymore, indicating an H. pylori infection. So those are the general treatments and the general diagnosis. Now, there could be neurological damage that could result from H. pylori infection, unfortunately, like Alzheimer's disease, Parkinson's disease. Another one, especially as women, you need to be watchful for this, are iron deficiency anemia and vitamin B12 deficiency. Uh, there could be a hepatobiliary system issue that could come up, like non-alcoholic fatty liver disease or hepatic carcinoma, which is a type of cancer, and dermatology, that means in the skin. So rosacea, rosacea. I don't know what it is, but anyways, just reddish pinkish skin. And so if you have like skin issues a lot, you might have H. pylori. If you have gastric cancer, you might have H. pylori. A peptic ulcer disease, which is in the stomach, but it's a different kind of ulcer, I think, that affects the specific portion of the stomach. There's gastritis, which is persistent inflammation, and this could be induced by the H. pylori as well. What I have here is this is the urease enzyme test, which could be used to find out We have H Pylori. These are all the different tests as well. So there's a stool one, and I got this from a paper I was reading. So let me see if the paper is still here because I don't want to steal their work, but I forgot to properly credit them. Was it here? Yeah, this one was very useful to making this presentation. This paper by Helena Cletus Sharandoma and Ifini Elibi Mimba, which is from this university in Nigeria. And this one was massively helpful. Even the references inside of it were very helpful. This was by Qifeng Sun, and Hupon Song is a chief writer, I think, maybe. This is the Hunan University of China, so a pretty big university, I believe. And they also included the part with traditional Chinese medicine, which I found pretty interesting to read. So thank you guys for writing these papers. They've been really helpful for helping me kind of understand and make sense of this. Um, yeah, until the next video, take care.